When you were little, you were probably told to learn from your elders. But what can we learn from the simplest of organisms that are capable of aging? Well, here's some life advice from bacteria. Hi, I'm Audrey Proenca, postdoc at PUCRS Brazil, and I'm here to talk about my work while at the Chao Lab at the University of California, San Diego. So let's get started with something cuter than bacteria. In mammals, it's pretty easy to spot the aging phenotype. As we age, we show a progressive fitness decline as we accumulate damage of various forms. What's most important, though, is that we retain this damage to produce healthy and rejuvenated offspring. But how can bacteria do something so complicated? Well, bacteria also accumulate damage, mostly in the form of misfolded proteins. These tend to clump together at the cell poles and can become too large to diffuse throughout the cell. When the cell divides, one of the daughters inherits this damage along with an old cell pole. The opposite pole has very little damage because it was just synthesized at the time of division. That is called the new pole. Next generation, the cell inheriting the maternal old pole inherits most of its damage, and it's called an old daughter. The sibling inheriting the mother's new pole is called a new daughter. This asymmetry has physiological consequences. So when Stewart and collaborators looked at bacteria inheriting either old or new poles generation after generation, what they found was that old daughters aged with every pole inheritance, growing more and more slowly, whereas the new daughters rejuvenated, they grew faster with each pole inheritance. However, a few years later, when people grew these same bacteria in devices that kept them healthy and well-fed for hundreds of generations, what they saw was that old lineages had remarkably stable growth over time, so they did not show a fitness decline and instead showed replicative immortality. So how can we reconcile the ideas of aging and immortality in the same system? To address this question, we grew bacteria using microfluidic devices. These devices have microscopic features that trap bacteria throughout the experiment while providing them with fresh culture media. So for these experiments, we used two different designs. One was the mother machine, which keeps old lineages over time, and the other was the daughter device, which keeps a 2D colony of up to 300 cells. So we looked at these populations of E. coli and followed their doubling times over time. First, for lineages consecutively inheriting the old poles, we also found that they had stable physiology. However, something else that we saw was that lineages inheriting the new poles every time were also stable, just with a lower doubling time because they're growing faster. Another way to look at this data is to plot these doubling times on a phase plane of the maternal values against the doubling time of the daughters. If there is physiological asymmetry between the siblings, then we expect to see a separation between populations on this plot. And from the real data, this is exactly what we found. Even in the absence of stress, old daughters were growing more slowly and new daughters were growing faster. But another interesting prediction arises from looking at this plot. Let's look at just the old daughters for a second. By tracing the linear regression between the doubling times of the mothers and the daughters, we end up with a slope that intersects the identity line. This intersection is predicted to work as an equilibrium point to which lineages will converge over time. How does this work? Well, let's say we are starting with a mother that has a longer doubling time, around 34 minutes. If we look at its daughter, it will have a predicted doubling time of about 27 minutes. Now, when this cell divides, if we keep on following the old lineage, its daughter will have a doubling time of around 25 minutes. Now, this next daughter will take around 24 minutes to divide. So with each division, you're getting a little bit closer to this predicted equilibrium point. And the same thing happens if we start with an old daughter that has a faster doubling time, let's say about 16 minutes. Each daughter will have a doubling time that is a little bit longer until they reach the equilibrium point. So if lineages in a real experiment stay constrained around this equilibrium point, that could explain why they look physiologically stable when you're looking at their growth rates or doubling times over generations. And here's how that worked. By following 60 generations consecutively inheriting old poles, we saw that they continuously return to this predicted equilibrium, although stochastic divisions can happen in which they go away for a second. And now we can push this prediction a little bit further and say that the same should happen for the new daughters. For each population, we should have two separate equilibrium points, one for the new lineages and one for the old lineages. So we looked at large populations growing in the daughter device and looked for lineages inheriting either new or old poles consecutively for at least 5 generations. And we found the new and old lineages were constrained around their respective equilibrium point. So we saw that these 2 points of equilibrium are stable for lineages continuously inheriting the same pole every time. But what happens when we start switching? For instance, let's say we take an old daughter that is born from a new mother. Well, we tried it, and we found that an old lineage born from the new equilibrium will age with each pole inheritance as it approaches the old equilibrium. And the opposite happens if you take a new lineage that is born from the old equilibrium. With each new pole inheritance, it will rejuvenate and get closer and closer to the new equilibrium. And so we end up with a deterministic age structuring of the population in which 2 points of physiological equilibrium are connected by processes of aging and rejuvenation. But all of this has been for unstressed populations. What happens to the equilibrium when you add stress to the environment? To answer this question, we applied extrinsic damage to our bacteria, mostly in the form of photooxidation. The overall elongation rates got slower and slower as we increased the levels of extrinsic damage. But most importantly, we found that the equilibria behaved differently. As the levels of oxidation became lethal, old daughters lost their equilibrium and stopped replicating, while the new daughters continued to divide. And finally, we asked whether this differential mortality has any implication for antibiotic survival. So we all know that antibiotic resistance can be a problem among bacteria, but another important issue is antibiotic persistence, in which susceptible bacteria can survive the treatment due to their very slow growth or dormancy. Can these persisters be found more often among old daughters, since they already tend to stop growing? Now, although persistence is easily found in batch cultures, it occurs to about 1 cell in every 10,000, which is a huge problem for single-cell microscopy. We tried a few different methods to increase their frequency in exponential populations. Something that worked was to induce growth arrest with a quick blast of tetracycline followed by a treatment with ampicillin, and we obtained a persister frequency of 10%. Although we looked for survival in over 1,000 lineages, because of imaging limitations, we could only simultaneously track the growth of 28 of them. We tracked these lineages as they arrested division, following a total of 78 new and old daughters, as most of them lysed due to the treatment. After a few hours of recovery, the only 3 cells that resumed growth were old daughters. To look further into it, we needed to track more cells at once, so we tried a different approach. Remember when I mentioned the protein aggregates could become trapped at the old cell poles? Well, we used those aggregates as fluorescent markers to indicate which cells were old daughters. This allowed us to inoculate stationary cultures onto agar pads with ampicillin and follow the survival. From this, we saw the classic biphasic killing curve that is a hallmark of persistence. By looking at the protein aggregates, we found that those cells harboring damage on the old poles yielded a higher persister frequency than the cells without damage. This could suggest that old daughters have a higher probability of persistent antibiotic treatments, which agrees with previous studies showing a correlation between oxidative damage and persister formation. To briefly summarize all of this, we found that new and old daughters have a deterministic asymmetry that creates stable age structures within the population. This stability can be disrupted by extrinsic damage, in which case the old daughters tend to stop growing first, whereas the new daughters can continue to divide. This differential survival also leads to a differential antibiotic survival, in which case the old daughters, because they stop growing, become persisters upon antibiotic treatment, whereas the new daughters tend to be more susceptible. And with that, I'd like to thank the organizers, all the members of the Chao Lab who contributed to this work, and thank you so much for listening.